Welcome to the Foundation Fighting Blindness National Chapter webinar on gene therapy. We are joining us today, and we have a wonderful panel of experts that will be presenting to us. My name is Davida Luehrs. I serve as a national trustee, as well as the president of the Washington, D.C. metro area chapter, a recent merger of the Northern Virginia and Montgomery County chapter. In 2005, the Foundation Fighting Blindness started the Northern Virginia chapter to help provide support and helpful information, resources, etc., to those dealing with vision loss. Since then, we have some amazing accomplishments that I would like to share with you today. In 2006, we hosted our first Vision Walk. We were one of the first 5 inaugural chapters to participate in the Foundational Signature Program. In 2014, celebrating our 8th anniversary, we were thrilled to celebrate a wonderful milestone of $1 million raised that year. And last year, celebrating our 15th anniversary, raised and surpassed $2 million. We have also hosted over the years over 40 chapter socials, which includes 6 dining in the dark dinners and 4 dancing in the dark galas that we created and developed. We were also the first chapter to host coffee and potluck socials. We could not have done and accomplished without an amazing team of dedicated volunteers. And I'd like to just take a moment to thank those people now. Kelly, Carol Edwards, Martha Jo Adams and her husband Dale, Fred Behrens, and we were later joined by Earl O'Donnell Khalsa. All have made significant contributions that have brought us to where we are today. I'd also like to thank the Northern Virginia Lions, who have contributed over $150,000 to our walk efforts. Because of COVID, we are not hosting the Dancing in the Dark Gala this year, but we are so excited to put our energies and our support to the upcoming February 28th Hope from Home event. It is now my pleasure to introduce and welcome Ben Shaberman. Ben is the Senior Director, Scientific Outreach and Engagement. For 16 years, he has been providing written information on retinal research advances for Foundation Fighting Blindness, print and electronic publications. He has provided multiple staff trainings as well as to constituents. He presents to local chapter events and national events. And he also enjoys working one-on-one with constituents and is very helpful in helping those individuals understand their particular disease and potential research therapies that may be of benefit to them in the future. I the mic over to Ben and welcome him, and he'll now give us an overview on today's program.
Great, Davida. Thank you so much for that intro. And thanks to you, Davida, your DC chapter, and all the chapters and grassroots efforts around the country and really throughout the world that make the research possible. Everything you'll be hearing about today, everything that we do is ultimately made possible by the hard work and generosity of those of you in the FFB family. So thank you. And it's a real privilege and an honor to communicate the research, to be the messenger, which is my role. And I feel very delighted to have such a great expert panel to talk about gene therapies. After I give you a brief warm-up and introduction to gene therapy, we'll hear from our panelists. They'll give you some presentations on their efforts. I'll ask them some questions, and then we'll open it up to Q&A We're going to get a ton of questions, and if we don't get to your questions today, we will answer them through email at a later date. So let's get started here. And actually, before I launch into my talk, I want to thank our generous sponsors who make this webinar possible and many of our other outreach activities. Our visionary champion partner is Genentech. Our outreach gold partner is AGTC. Our outreach silver partners are Apellis and Astellas. I like the alliteration there. And also outreach silver partners are Biogen, Mira GTX, and Spark Therapeutics. So before I give a basic introduction to what is gene therapy, I think it's very important for us to understand the multiple applications for gene therapy. Most of the time when we think of gene therapy, we think of gene replacement or gene augmentation, where you're replacing the mutated or bad gene with a good gene. As most of us know, retinal diseases, inherited retinal diseases, are caused by mutations in a single gene. And in many cases, if you can replace that mutated gene with a good gene and you have some viable retina left, it can be an approach to saving and possibly restoring vision. But in many cases, there may be other applications for gene therapy that are more appropriate. And those applications include neuroprotection, where you're basically trying to just preserve photoreceptors independent of the mutated gene. Another application is targeting a pathway to relieve a symptom or to inhibit damage. That too is often gene independent. And optogenetics is a way to restore vision to a retina that's lost all its photoreceptors by harnessing other cells in the retina. And we'll hear Dr. Flannery wax poetically about that option. I'm going to cover these options in a little more detail in a few slides. But to start off, I just want to give a basic introduction to what a gene is and why it's important. And for me, the best analogy for a gene is to think of it as like a recipe. And our cells have a complete set of genes, somewhere in the neighborhood of 20,000 genes. And our cells read the genes. They read them much like we would read a recipe, so they know how to make proteins. And it's, at the end of the day, it's the proteins that are really important to the health, well-being, and function of our cells. The recipes are the instructions. Now, these genes or recipes are comprised of nucleotides, which we represent as letters, A, T, C, and G being the fundamental letters. And we have somewhere in the neighborhood of 6 billion of these letters in our complete genome. This is in every cell. So our genome is pretty complicated. But when there is a mistake that leads to a problem, a misspelling in this code, in this recipe, in general terms, two things can happen. We don't have enough protein made in the cell, or the wrong or a toxic protein is made. And that's when there can be retinal degeneration and vision is lost. So on this slide on the left, I have an image of a protein. This happens to be RPE65. RPE65 is the protein that helps metabolize vitamin A. Vitamin A that we normally get through our diets it helps make our retinas light sensitive. We need vitamin A to see for our photoreceptors to function. And this image is sort of like a bird's nest of coils and strings. It's a relatively complex entity, a collection of amino acids. So I just wanted to give you an appreciation of what a protein is and what it looks like. And again, it's the proteins that do the heavy lifting in our cells. So when we're talking about gene therapy, the question is, how do we get genes to retinal cells? And the way that that's done in the current world, in most clinical trials and many preclinical trials, is by human engineered viruses. And the most commonly used virus these days is called an adeno-associated virus. And I think a good way to think of these viruses as like vast container systems. So a gene therapy is like a ship or a truck that contains all these viral containers. And these containers are loaded with the therapeutic gene. So the image on the right here, this knobby, colorful ball, is a shell of a virus. It's a capsid. And that's the container that you put the therapeutic gene in. So there are literally billions of these containers in each gene therapy. And we use these AAVs, these adeno-associated viruses, because they're relatively safe. Most of us are naturally exposed to these, and they're benign for the most part. And they're pretty good at penetrating retinal cells to deliver the cargo. And researchers like Dr. Flannery and Dr. Boye are finding ways to make them penetrate the cells even better. So on this slide, on the left, I show how you get the gene therapy actually into the retina. And this image, the needle sort of ominously going through the eye underneath the retina, is a subretinal injection. And this is the most common way that gene therapies are administered in clinical trials right now. And it's the injection of a tiny drop of liquid underneath the retina. And for those of you that can see, the injection kind of lifts up the retina. So this is not an uncommon procedure, but it's also not trivial. It is a surgery. And for people with retinal diseases, the retina is delicate, so we need to be really careful when we do subretinal injections. There are alternative ways emerging to deliver gene therapies, including delivering them in the vitreous in the middle of the eye and underneath the sclera through suprachoroidal delivery. Those hold promise for being less invasive. So on the right, I simply show just one viral container containing, again, a therapeutic gene penetrating the retinal cell to deliver the therapeutic cargo. So that's how we get gene therapies into the retina. So let's talk about that first application, gene replacement. And I'm going to rifle through these trials and preclinical studies. There's a lot here. This is a partial list. This isn't everything. There's a lot that's moving toward clinical trials that I just don't have time to cover. But gene replacement is by far the most advanced form of gene therapy right now from a clinical standpoint. So most of us, I'm sure, are aware of Luxturna, the first FDA-approved gene therapy for the eye or an inherited condition. It's for people with RPE65 mutations, which cause severe vision loss and LCA. They can also cause RP. FDA approved in 2017, more than 3 years ago. And this treatment has had dramatic vision improvements for many people who are receiving it. Kids are putting away their white canes. They're seeing the faces of loved ones for the first time, even seeing stars in the sky. So in addition to this Spark Therapeutics gene therapy now being available, it's really paved the way for the development of other gene therapies to treat inherited retinal disease. It's really provided affirmation to companies and researchers that gene therapy is a great way to go to address these conditions. So other trials that are underway include an X-linked RP trial being conducted by AGTC. Their gene therapy is currently in a phase 2. They've had vision improvements, good results. They're planning for their next phase, phase 3. AGTC also has an early stage clinical trial underway for achromatopsia, which causes extreme light sensitivity. The condition does. That's still at an early stage, some early encouraging results, and we're waiting to hear more on that trial. Mira GTX, along with their partner Janssen, also has an X-linked RP gene therapy clinical trial underway. They too have had good results in their phase 2, some vision improvements, and they are also planning their phase 3. They too have an achromatopsia gene therapy clinical trial, or actually a couple underway, that are still at an early stage. And we'll get to hear a lot more about, especially the XLRP gene therapy trial, from Dr. Adamson later in the talk. Biogen has finished recruiting for their choroideremia gene therapy clinical trial. Some good results leading up to that phase 3. So when the phase 3 is completed, we'll get results there. If those results are promising, then I'm sure they'll move to seek regulatory approval. Biogen as well is conducting a phase 3 clinical trial for an XLRP gene therapy. They had some encouraging results in their earlier trials for that. At a preclinical stage, Biogen is also working with Mass Eye to launch a gene therapy trial for PRPF31 mutations, which are a relatively common cause of RP. Now, the company 40 Molecular Therapeutics is a little newer to this space. They have early stage gene therapy clinical trials for choroideremia and X-linked RP underway. Their approach is a little different. They are doing intravitreal injections, that potentially less invasive approach. So they're still at an early stage. We look forward to results from them. Harama, French company, has a PDE6 beta gene therapy clinical trial underway for RP. No results reported for that yet. They're collaborating with Leiden University Medical Center in the Netherlands to advance a gene therapy for CRB1 into a clinical trial. Novartis has a gene therapy clinical trial underway for RLBP1, which lead to mutations for which can lead to RP. Another company, Iveric Bio, has a lot of great preclinical work underway for retinal disease gene therapies, including for rhodopsin, which is associated with RP, CEP290, an LCA gene, Best disease, and USH2A. And we'll get to hear about the great work of Atsena Therapeutics and Shannon Boye. They recently launched a trial for LCA caused by GUCY2D mutations. And Shannon is also doing some great preclinical work on gene therapy for USH1B. And finally, the nonprofit Odelia is teaming up with Mass Eye to advance gene therapy for RP GRIP1 mutations, which cause LCA. Again, a lot of other gene therapies that are moving toward clinical trials. And I just wanted to mention ABCA4 is an important target. That gene, when mutated, is the most common cause of Stargardt disease. So let's talk briefly about the other 3 applications for gene therapy. And number 2 on the list is neuroprotection. And with neuroprotection, we're simply trying to save photoreceptors, stop or slow progression of disease and vision loss, independent of the mutated gene. And we have been funding the Institut de la Vision in France for many years to develop a gene therapy that's targeting cones. And what these researchers at the Institute found is that rods secrete a protein that helps keep cones alive. If you have RP, you may be well aware that when you lose your rods, the cones can follow. And the Paris researchers hypothesized that it's this protein that if you can secrete it, you can actually save cones. So they aptly named this protein rod-derived cone viability factor, and they are hoping to get a clinical trial off the ground this year, later this year, I should say. And they'll be targeting RP and perhaps Usher syndrome, because Usher syndrome is RP with hearing loss, and potentially other conditions where saving cones is important. And we should remember that cones are important for vision in that they provide central vision, visual acuity, color perception, and the ability to see in lighted conditions. So if we can save a few cones, we can save meaningful vision for most people. And the company that's doing this, I should add, that came ultimately out of the Institut de la Vision and some other groups is Sparing Vision. This is a company funded by the Foundation's RD Fund, our venture philanthropy fund. So the next application, targeting a pathway to inhibit a symptom or damage. And we're going to talk a little bit about AMD first, so you can understand how gene therapy is being used to address AMD. AMD is a little different from an inherited retinal disease in that there are multiple factors that can cause the onset of AMD. It's not a single gene disease. There are lifestyle factors like smoking and probably diet, sun exposure. Unfortunately, as we age, our risk of AMD increases. But about 15 years ago, researchers learned that our innate immune system, specifically the complement system, is a factor in the onset of AMD. Most of the time, we want our immune system to function well so it can fight off pathogens and bad things. But if it's overactive, it can cause damage. So the idea behind some gene therapies is to mitigate or temper the immune system so that damage doesn't occur. And there are a couple of companies currently in clinical trials for gene therapies that are targeting the innate immune system, the complement system. And those include Gyroscope and Humira. So stay tuned, they're still at an early stage, but this is an interesting and promising way to address AMD. Now, as many of you probably know, sometimes AMD migrates from the dry form to the wet form, where you have the growth of these leaky blood vessels, and those leaky blood vessels lead to death of retinal cells and photoreceptors. And we have FDA approved treatments to address those leaky blood vessels, but those treatments have to be administered to the eye on a regular basis for the rest of the patient's life. So those injections, the trips to the doctor's office, not convenient. So companies are developing gene therapies that are one-time approaches to inhibiting the growth of these leaky blood vessels. And there are 3 companies that I have listed in this space: REGENXBIO, Adverum, Humira. They're all in clinical trials. And actually, REGENXBIO is a little further along. They're moving toward a phase 3, and they've had some encouraging results in their earlier stage clinical trials. And then finally, the 4th application, again, is optogenetics. And so optogenetics really applies to people who have lost virtually all of their vision, who perhaps have light perception or not even that. And what happens in an advanced retinal disease where you've lost all your vision is you've lost all your photoreceptors. And a lot of the approaches I've talked about won't be appropriate because there aren't any photoreceptors left to save. So with optogenetics, what researchers are doing is they're targeting other cells in the retina that survive after photoreceptors are gone, namely ganglion cells and perhaps bipolar cells. And they're using these optogenetic therapies to bestow light sensitivity to these ganglion cells and these bipolar cells. And again, Dr. Flannery is going to give you a lot more insights into how these work and some of the pros and cons. There are actually 3 clinical trials underway for this approach. Some involve the use of goggles or glasses to enhance or boost the visual experience potentially. But I will say that all of these are still at a relatively early stage and we don't have results. Optogenetics is still pretty new and we're still learning a lot. And there are limitations right now to what we can do with optogenetics in terms of the potential to restore vision. Now, Dr. Flannery's work is a little more advanced. It's still at a preclinical stage, but we'll hear more about that. He's a scientific co-founder of Vedere, again, an RD fund company, part of our venture philanthropy portfolio. And he will talk more about their work, I am sure. So finally, to finish up, I just wanted to review some opportunities and challenges in gene therapy for IRDs. One great thing about gene therapy is one injection should last many years for a patient, maybe even their lifetime. So far, so good. We've got a good track record, especially for AAVs. They've been relatively safe and we've had some good efficacy in some of the trials. Some of the challenges is that the genes can be too big for our AAV delivery systems. Genes like USH2A, MYO7A, ABCA4, they don't fit into that standard viral AAV container. Another thing that I haven't addressed at all, and our panelists may address this, but sometimes when we're doing gene replacement, we also have to knock down the bad gene because it's making a toxic protein. And that means in those cases, gene replacement is actually a two-step process, replacement and knockdown. And that can be the case in dominant diseases. Again, we're working to find better ways to inject these gene therapies. Subretinal injections work. They're not trivial, and maybe intravitreal will be a better option. We'll be hearing more about that. And one thing I didn't cover at all, just for sake of time and being brief, is our gene editing and RNA therapies, which aren't our standard gene replacement therapies or delivering a whole gene, but they are promising approaches that are in clinical trials and have some important applications. So with that, I just wanted to say thanks for letting me make this brief introduction. For more information, you can always go to fightingblindness.org, where we cover a lot of what I've talked about. Clinicaltrials.gov has great information on all the clinical trials underway in the US and many overseas. And everybody should be in myretinatracker.org, our patient registry, so that you are on the radar screen of researchers and clinical trial recruiters. So with that, I would like to begin our panel presentations. And we're going to start off with presentations by Doctors Boye, Flannery, and then Adamson. If we have time, I'm going to ask a few canned questions, and then we'll go to Q&A from you, the audience. So Shannon, feel free to share your screen. I have relinquished control. And while she's doing that, I'm going to give a brief introduction to Dr. Boye. She is an associate professor and associate chief of the Division of Cellular and Molecular Therapy in the University of Florida's Department of Pediatrics. She's a scientific co-founder of Atsena Therapeutics, which is a company funded again by the Foundation's RD Fund, our venture philanthropy fund. And she's been a longtime recipient of Foundation's grants, great investments for us. And she's also a member of our scientific advisory board. And I'll add that Dr. Boye is a little under the weather. So thanks for being a trooper and being with us today, Dr. Boye.
Thank you, Ben. Good morning, good afternoon, everyone. I'm sorry if I sound a little froggy this morning. Great intro talk, Ben. That's really going to help with my presentation. So As Ben mentioned, I'm an associate professor at the University of Florida, and I've spent over 15 years now developing gene therapies for various forms of inherited retinal disease, as well as focused on the AAV vector itself. And so my team and I, which includes my husband, who's also a co-founder of the company that I'll be talking about, we developed these technologies, and over the course of our careers, we would out-license them to a variety of companies, small companies, medium companies, all the way up to big pharma. And something that we noticed is that, it was really hard to hand off a technology that you had worked on for so many years to a group of people that, even though they were in big pharma, they certainly didn't have that depth of experience that you had in the lab working for so long on something. And so we sort of saw the good, the bad, and the ugly being, of course, when business decisions overrode science decisions. So we both reached a place in our careers where we felt that it was important for us to form our own company and have a little bit more control over the scientific direction that those projects took. And so that really forms the basis for Atsena Therapeutics. And we were very fortunate in the spring of 2020, amidst a global pandemic, so this was all done via Zoom, to raise our Series 1 funds. And the RD fund of the FFB was absolutely instrumental in making that happen. Hatteras Venture Partners was our other lead investor. We had several other minor investors. And then we were very excited in December of 2020 to close out the year with our Series A financing from 3 additional investment firms. And I can't thank these guys enough, because of course, getting all of these programs over the finish line requires capital, not just science and bright minds and elbow grease, but you need money to get it done. So I really appreciate the RD Fund and everybody else who's helped in this effort. So just a little bit about our team, and I don't mean for this to be a commercial for my company, because really the programs that we're working on have been things that we've worked on in the lab for a number of years. It just so happens to be in the company now, but I think it's in good hands. We have a fantastic CEO, Pat Ritchell, who has a lot of experience in this space, including being a founder of a former founder of another AAV gene therapy company, also very active in the rare disease community. Our COO is Nasser Hassan, who has experience both in small gene therapy companies and big pharma. Christine Kay, who's a very active member of the FFB family, is a vitreoretinal surgeon who's actually doing these surgeries on inherited retinal disease patients all the time. Mike Kelly, who has a depth of experience in manufacturing these AAV vectors. As I mentioned, I co-founded the company with my husband, who's currently serving as the Chief Technology Officer. And we're also very fortunate to have a stellar clinical advisory board to help us walk through our clinical program and get us ready to bring our preclinical programs into the clinic. And I just want to mention, for the visually impaired members of the audience today, there are images and slides, but I'm basically going to describe everything. So hopefully you won't be missing anything over the course of the presentation. So we are at Atsena a leading ophthalmic gene therapy company with both clinical and preclinical assets. Our clinical asset is Leber congenital amaurosis type 1, which we'll talk about more in a moment. But this is essentially a homecoming story, because I started working on this program in 2004. And as I said, we out-licensed technologies over the course of our careers, and one of them was the LCA1 program. We actually licensed it to Sanofi, which is a large company. But they decided at a certain point that they weren't interested in ocular gene therapy anymore. So they started looking for someone to license that program back to. And so we were very fortunate that with the help of the FFB, that we were able to bring that program back home. And I'll talk a little bit about it in a moment. We have 2 rapidly advancing preclinical programs, one of which is undisclosed. It'll probably be disclosed later this year. But the other is for the myosin 7A form of Usher syndrome, or USH1B, which is also based on many years of research in my lab. In addition to the clinical programs and the preclinical programs, we're also heavily focused on our technology platform. In other words, developing bigger, better, faster, stronger AAVs with which to conduct these gene therapies, so that we can ensure that these treatments are very efficient and, more importantly, very safe in patients. Okay, so just a little bit about our clinical program, LCA1. This form of LCA is not caused by mutations in RP65, which is of course probably the most famous form of LCA. This form of LCA is caused by mutations in Gucci2D, and this form of LCA actually affects about double the amount of patients that have the RP65 form, so it's very common. The gene encodes a protein, or it makes a protein. In other words, the gene is a recipe for retinal guanylate cyclase, which is expressed in photoreceptors. And put simply, when photons of light hit your photoreceptors, it initiates this cascade of events, this party of proteins all getting together and doing something to make sure that you can see. But retinal guanylate cyclase 1 is involved in shutting down that cascade. So it can't be on all the time, or else our brain won't be able to interpret anything as vision. So if you don't have RepGC1 there, your photoreceptors are essentially stuck in the light. And that's sort of the underlying basis for this disease. But despite the profound dysfunction in these patients, they're typically diagnosed within the first few months of life. Their retina structure is maintained over their lifetime. So this is pretty different than other forms of LCA. But what that means for us is that if the cells are there, then we can easily perform gene supplementation and hopefully restore function to those cells. So that's the goal. And as I mentioned, we've been working on this condition for about 15 to 20 years now. And this is that entire span of my life on one slide. But what we've shown in multiple mouse models of this disease is that we could fully restore useful cone and rod mediated vision to those animals, whereas the untreated mice were completely blind. We show that we can drive expression of that really important GC1 enzyme in the photoreceptors where it's supposed to be. We also show that we can significantly improve retinal function as assessed by an electroretinogram, or ERG. We can prevent photoreceptor degeneration. We can restore normal levels of the enzyme as well as its biochemical partners to the cell. And put simply, this mountain of proof of concept data had us feeling pretty confident that we should move this forward into the clinic. In terms of where things are now, I can't say much, of course, because it's under a company umbrella and I don't want lawyers to get mad at me. But we have finished enrolling cohort 1. That happened as of last February, and we have already started enrolling our cohort 2 patients. And we have very early signs, encouraging signs of both safety and efficacy in these patients. So things are going very well. So on to our preclinical programs. As I mentioned, one of them is not disclosed yet. It will be later this year, but the one we have disclosed is a form of Usher syndrome caused by mutations in myosin 7A. Very much like LCA, Usher syndrome can be caused by a number of different gene mutations, but this form is one of the most common. It affects about 25% of Usher's patients. And it's a particularly aggressive form, because patients are born profoundly deaf. They start to lose vision within the first decade of life, and they also have vestibular defects. And that's because the protein product that myosin 7A makes is expressed in the inner ear as well as in the retina. So the way that patients with USH1B lose vision is in a very retinitis pigmentosa-like fashion. So in other words, their peripheral visual field is lost as a result of the death of the rod photoreceptors, and ultimately the patients develop tunnel vision. But the really good thing is that patients can maintain this central island of healthy photoreceptors well into even their 4th and 5th decade of life. So this offers us the opportunity to provide that healthy gene to those living cells that are still remaining there. As Ben alluded to earlier, though, one of the hurdles with USH1B and with myosin 7A is that this gene is too large to fit within a standard AAV vector. So we have licensed technology from my lab into Atsena to overcome this hurdle. And it's really it's a very simple concept when you think about it, even though it's kind of, very involved science. But what you simply do is you take the large myosin 7A gene, and you can do this with other large genes as well, and you divide it in half. You put the front half of the gene in one AAV vector, and you put the back half of the gene in another AAV vector, and then you co-deliver those 2 vectors to the same cell, in this case, the photoreceptors, for instance. Those 2 gene halves are released from the vectors, and they find each other via a region of complementary sequence. So they sort of bind to each other. And then they recombine to form the full-length myosin 7A gene, and then go on to make the full-length myosin 7A protein. So we've been working on this for a number of years, and we've shown that this works very well to produce full-length myosin 7A, both in cells in a dish, in subretinally injected mice, and in subretinally injected macaque. We show that dual AAV myosin 7A vectors are capable of restoring one of the retinal phenotypes in a mouse model of USH1B. We also have unpublished data showing that our dual vectors can restore balance to a mouse model of USH1B, which is another exciting avenue that we'd like to explore. We also showed that when those two gene halves come together, that the message they make, or the transcript, which is sort of the intermediate step between gene and protein, is 100% normal. And we've also shown that these dual vectors are well tolerated in macaque. So we're very excited to bring this program forward with Atsena. So those are our clinical and our preclinical programs, and I just want to talk for a few minutes about our capsid technology. So of course, we are focused very heavily on subretinal injection now because, as Ben mentioned, that's the most efficient way of getting genes or gene editing reagents, et cetera, to cells of the outer retina. And that's where most inherited retinal disease starts, is in cells of the outer retina. But we are also focused on developing AAV capsids for intravitreal injection, simply because it's much easier to deliver vectors via this route. So just briefly, I want to talk about some of the vectors that we've been looking at for subretinal injection. What you can see, and I'll walk through, if you can't see this, what is on the screen. So just some background. It's been shown before in clinical trials, subretinal injection is a relatively invasive surgery, and it doesn't happen in all patients, but in some patients, if you detach the retina underneath the fovea, where those very precious cones are, you can exhibit a loss of central retinal structure, and in some patients, even a loss of visual acuity. This isn't common, but it is seen. And so when we saw that as a field, everybody rallied together and tried to come up with ideas to more safely target the foveal cones without actually having to detach the fovea during the subretinal injection. And so we've identified an AAV vector that can be subretinally injected outside the fovea. So on the top right of this screen, you can see an OCT scan. This is a scan through the patient's retina, which shows you that the fovea was not detached during the injection. And let me back up. This is not a patient. This is a monkey. And so we delivered 3 subretinal injection blebs, again, not detaching the fovea. And then we looked for expression of the AAV-mediated protein, in this case, a GFP, or green fluorescent protein. And we looked in the fovea, and we found that 98% of the foveal cones were infected by that virus, even though the subretinal injection did not go underneath that cone-exclusive fovea. So we're really, really excited about these vectors because, again, they afford us the efficiency that you get with a subretinal injection, but they also afford us the ability to more safely target the foveal cones. And so this is the vector we're considering using for our USH1B program, as well as other programs that we're looking at. But more than that, it will be very useful for other indications that require a transduction of a wide area of retina, because these vectors are laterally spreading, and especially useful for conditions where you want to very safely target those foveal cones. Excuse me. Okay, just briefly want to discuss some of the vectors that we're looking at for intravitreal injection. So what we did a few years ago was we did something called a capsid library screen. And you can think of a library screen as a billion different AAV capsids that you're going to throw against a wall, and then you're going to ask what's stuck, okay? So our goal was to see which AAV capsids in that library were best at getting to the photoreceptors of a monkey following intravitreal injection. So in order to ask that question, we needed a monkey that had photoreceptors that we could pull out at the end of the experiment. So we did that by doing a pre-subretinal injection with AAV5 GFP to exclusively label the photoreceptors. So put simply, we made the monkey's photoreceptors green. We then made their ganglion cells red by doing another procedure, which I won't get into today. But essentially now we have monkeys walking around with green photoreceptors and red retinal ganglion cells, into which we can inject a gabillion different AAV capsids. At the back end, we can sort out the red cells and the green cells and ask which AAVs got into them. And so we found a number of AAV variants that were heavily enriched in this screen that outperformed benchmark vectors. So what you're looking at on this screen here are images of mouse retinas that received intravitreal injections of our new AAV capsids. And you can see that many of them are driving much stronger protein expression than our benchmark vectors, which are on the far left and on the far right. So we were really excited about the increased potency of these vectors that we saw following our experiment.
This is a national webinar and we may have different participants. And
I think someone needs to mute themselves. I hear a voice. So then we took the most potent capsids from our screen and we tested them individually in intravitreally injected monkeys. And to make a very long story short, we saw about a 4 to 10-fold improvement in the transduction of monkey retinas with these new capsids over benchmark vector AAV2. Something really important to mention here is that we used a very clinically relevant dose of AAV. So 1 times 10 to the 11th total vector genomes delivered was what we used. And this is very close to what Adverum is currently using in their wet AMD clinical trial. So our goal was not just to show really bright GFP expression in these monkeys. We wanted to look at something that was clinically relevant that ultimately would be a dose that we would use in patients and not a dose that would cause inflammation. At our high dose, you can see these are sections through the foveas, the cone-exclusive foveas of all of the monkeys, and we see a very nice pattern of expression in the ganglion cells in the inner retina, some Mueller glia. But the important point here is that we're not seeing any signs of inflammation in those monkeys, which is important. Again, we wanted to do this at a clinically relevant dose. When you compare this to some benchmark vectors, you can see that we're achieving approximately similar levels of transduction using 50-fold less vector. So we do think that we're moving in the right direction. But one thing that had us scratching our heads was when you look at all of the mouse eyes that were injected with our novel capsid variants, this variant here, which we call P2V1, was actually the most enriched variant. So this was what we found in more primate photoreceptors than any other AAV capsid. But you'll notice that its ability to drive protein expression wasn't that much better than the benchmark vectors, right? It's not that much brighter, whereas these guys are super bright. So why was this the most enriched variant in our screen? So we got thinking about the way that we did our screening of that capsid library. And remember, I told you that in order to isolate the photoreceptors of those monkeys at the end of the experiment, we had to do a pre-subretinal injection with an AAV vector to make the photoreceptors green. And so we hypothesized that maybe doing that early subretinal injection primed those monkeys to have an antibody response against AAV. In other words, they developed antibodies that could neutralize future injections of more AAV. So we tested that theory by taking human vitreous samples from our ophthalmology clinic. So these are patients that were undergoing surgery, routine surgeries, and we saved a little bit of their vitreous. And we looked for the samples that contained a lot of antibodies against AAV. So those would be our inhibiting vitreous samples. We then incubated those inhibiting vitreous samples, in other words, those samples that had a ton of AAV antibodies, with either our P2V1, our lead variant, or benchmark vectors AAV2 or 7M8. And then we threw that combination onto cells and we asked, could those viruses still infect cells? And the answer for P2V1 was yes. So that sounded like a lot of scientific jargon, I'm sure, but the bottom line is that we found that our novel capsid variant is capable of evading AAV antibodies. So that gives it a huge advantage over benchmark vectors, and we see it outperforming benchmarks about 75% of the time. Why that's important is because if you're a patient that wants to enroll in a clinical trial involving intravitreal injection of AAV2, you may already have pre-existing antibodies against AAV because AAV2 is found naturally in the environment. About 70% of us have naturally occurring antibodies. So for indications where we want to use an AAV2 based injection into the vitreous, it would be very useful to have something that could evade those antibodies. So this could drastically increase patient eligibility in those types of clinical trials. So I will end here. I hope I've given you a good flavor for the things that have come out of my lab and now are in Atsena. Of course, we have our eye on other programs, but this is just a smattering of what we're doing now. But I just want to thank Ben Yerxa especially because he, through conversations at conferences and airports, really was very instrumental in holding my hand through initial company formation. I'm definitely not a business person. I'm definitely just a scientist. To Rusty Kelly, who's also at the FFB, who was instrumental in helping us reacquire the LCA1 program from Sanofi and help us bring it home. And to everybody on the webinar today for your attention, and everybody else in the FFB who's been so supportive of my career over the years. Thank you very much.
Great job, Shannon. Thank you for that wonderful talk. And one thing I want to emphasize that you sort of casually mentioned, you go through a slide and say, this is like a lifetime of work on one slide. And I think sometimes when we go through a presentation in 15 minutes that highlights a lot of work, we don't appreciate the years and years of painstaking research that takes place to get to this point. And it isn't a linear path. There are obstacles and setbacks. And so thank you for your great efforts, and congratulations on where you're at today.
Thank you for helping us to fund it.
Oh, well, our pleasure. And ultimately, we thank the people on this webinar for raising the money that helps make that possible. So I am going to go back to a couple of my slides as a backdrop for Dr. Flannery. And I think, Dr. Flannery, that is the slide you were wanting as the backdrop. And let me give a brief introduction to Dr. Flannery. He's been a faculty member in the Department of Molecular and Cell Biology at UC Berkeley since 1985. He's also Associate Director at the Helen Wills Neuroscience Institute at Berkeley. He serves as Vice Chair on the Foundation Scientific Advisory Board. We've been funding John for many years. I know you've been on our SAB for many years as well. And he is scientific co-founder of Vedere, which you'll be hearing about. And again, that is a company that has been funded by our Retinal Degeneration Fund, Venture Philanthropy Fund. So Dr. Flannery, the screen is yours.
Thanks very much, Ben. Thanks for the generous introduction. Yes, as Ben mentioned, I'm a professor at UC Berkeley since 95, not since 85. And I have a lab that's functioning and currently under COVID, and we have 8 graduate students and a couple post-PhD fellows. Each of them is working on a different project. And I think I'll use my time to give you some examples of those projects, because I think they touch on most of the topics that are in this survey slide that Ben has put up for us. And also, I've taken my time while Shannon was giving her eloquent talk to look at the Q&A, and you're now stacked up at 45 questions. So I think what I'll do also is I'll try to answer as many of the questions that you've posted in the chat with examples from projects that we're currently conducting. So one of the questions that comes up often is that why are you working on the diseases in the clinic, or why are companies working on the ones that they are working on and ones that they're not working on? Why are they not working on those? Another one that comes up very commonly is that how do you pick which ones you work on, or how hard is it, or can you find one that will work on all the different mutations? So every one of the current clinical trials that I'm aware of, which is over 10 or 11, which Ben and Shannon talked about, almost every single one of those now is working on what we call an autosomal recessive disease. That means that the defective gene was inherited from both of the parents. And that it also means that most of the time, as Ben showed you, is the defect is in a protein. And in these recessive diseases, what's happening is that your photoreceptor cells are not making that protein. So that missing part is what is put back with the gene therapy, as Shannon explained, is that the therapy for these recessive diseases, where you're missing a necessary part for your photoreceptors to survive, the therapy is to put the gene for a normal copy of what you're missing back in. But that also leads to one of the big questions is why are you picking the ones that you're picking and why are you not picking other ones? And if you look even more carefully, you see that sometimes there's 2 or 3 companies working on the same one, which really drives people crazy when they see that nobody's working on their family's condition. So the clinicians who are genotyping patients have been remarkably successful in finding the genetic underpinnings for these inherited diseases. And RP and these retinal degenerations are probably the most genetically diverse disease known to man. There's over 300 different gene defects we now have cataloged that all cause blindness. So it's very, very difficult to design a therapy that would work on all of them because the parts are very, very different. The analogy I like to think of, especially since it's Saturday, is that if I'm working in my 30-year-old car and it's not going to start, it'd be nice to just go to the auto parts store and get the part. But at the auto parts store, there's 100,000 parts. And if I get a new radiator, that's not going to help my car if the starter's broken. So the trouble with these recessive diseases is that the elegant repair, and Shannon gave you examples of that for 2 diseases, is that if you can identify by genetically screening the patient exactly the defect that's wrong, the best treatment is to put back a normal copy of that. So that would require companies now to make 300 different therapies, which is going to be very difficult. But making one therapy for all of them is probably not practical. This also explains why the price, the current price for the LCA2 therapy from Spark Roche, Luxturna, is $700,000 per patient. So the reason for that is that the cost of getting that treatment through all the stages of testing to make sure it was non-toxic was several $100 million. That therapy will only work for patients that have a defect in that exact gene, which Ben showed you the ribbon diagram of. It's the RPE65. So estimates are there's only 2,500 people in North America that have that particular gene defect. So one of the reasons why it's so expensive is that the hundreds of millions of dollars that it costs to develop that treatment is only going to be applicable for a few thousand patients. So the cost per patient is very, very high. One of the reasons for people trying to make more ubiquitous therapies that work for many, many patients is that if you could design therapies that work for large numbers of patients, the price would not be as high. For example, Stargardt's is the most prevalent IRD, and a lot of companies are interested in that for the particular reason that there's a lot more patients with Stargardt's. However, Stargardt's is biochemically a very difficult IRD to treat, so it hasn't been very successful. As Shannon described, the most common mechanism for treating recessive diseases is to use adeno-associated virus. And one of the great things about adeno-associated virus is that one injection should be effective for the life of the patient because the cells that it treats in the retina don't turn over. And so once the therapy is applied, it should be one and done. So that's really excellent. However, for dominant diseases, which are diseases that are very different than the recessive diseases I just described, about half of the patients that have retinal degenerations have a dominant disease where it's not that they're missing a piece that's required, they're not missing a protein, they're making a protein that has a defect in it. So it has the wrong shape, or it's only making up 2/3 of it, or it folds up in the wrong shape and doesn't make that beautiful ribbon structure that Ben showed. And so, as Ben mentioned, the therapy for dominant patients is going to be more difficult because the first thing the therapy has to do, it has to get rid of this defective protein, because that defective protein is toxic. It's killing the cells. And in many cases, after you design a way to get rid of it, then you have to do what we did for recessive diseases and put back a normal copy. So the gene therapy tools that Shannon and Ben described using AAV for recessive diseases, what we want them to do is to work for decades, because that disease progresses in the patient for many, many years, if not decades. So for dominant diseases, what we want to do is we want to get rid of this toxic protein. And the new technology for CRISPR editing is probably the way that that's going to happen. And so CRISPR editing will allow therapeutics to go in and to correct the defect that makes the toxic protein so that the patient will no longer make that. However, the CRISPR is very different. You can think of it as editing, like in the word processor, and the requirements for how you deliver it to the eye are going to be very different. Whereas you want the recessive therapy to last for decades, the CRISPR, when it does its editing, most people think it'll do the editing in a few hours to perhaps a day or two. Then you want the CRISPR to stop. You don't want it to continue looking around for something to edit. So the therapy for CRISPR has to be in some ways the opposite of what we wanted for recessive diseases. We want a therapy that goes in the eye, works for a short period of time, corrects the gene defect, and then stops. So we're doing a project with Jennifer Doudna's lab. She won the Nobel Prize recently for this development of CRISPR, to do that, to design short-acting editing for autosomal dominant diseases. So that's one of the projects that two of the students in the lab are working on, gene editing for dominant diseases. Another thing that you asked questions about are these gene-independent therapies and neurotrophins. And we're collaborating with Sparing Vision, Jose Zahel and Terry Laviard, to design the AAV vectors for RDCVF. And as Ben showed you, RDCVF, the rod-derived cone viability factor, is known to be a factor that is secreted from rods, but is required by cones to live. So to expand a little on the way Ben described it, that discovery by the group in the Institut de la Vision in Paris explained a literature for maybe 30 or 40 years that ophthalmologists were puzzling over. And patients know this very well, but it wasn't very well understood, is that if you have a genetic defect that kills your rods, most patients have good macular and foveal cone vision for decades. But at some point when they've lost somewhere around 3/4 of their rods and their tunnel vision is quite profound, then they start to lose their central cone vision, which is devastating. What people have found out since then is that those cones actually don't have a defect in them. What's happening is the cones are no longer able to survive because this factor, the rod-derived cone viability factor that was supplied by the rods, is missing. So the sparing vision therapy is to do an intravitreal injection of a AAV vector. It's the 7M8 vector that we developed quite a few years ago. It's an intravitreal injection. The idea is that if you can supply this secreted factor that the rods used to supply, then you may be able to keep the cones alive. And in patients that live in a city, having your cone central vision persist is probably going to be very beneficial for your lifestyle. So we're collaborating with Maureen McCall in Louisville. She has a large animal transgenic pig model that has a human RP gene in it. And we're doing intravitreal injections in the pigs, and the large animal studies in the pigs where we're delivering RDCVF show that the
answer to this one, but
in the pig model seem to be very well preserved. The therapy doesn't do anything to stop the rod cell loss, but it does do quite a bit at keeping the cones intact. So we think that's a good therapy because it doesn't require you to know what's wrong with the patient's rods. It works on preserving the cones regardless. So it's gene independent, and it should be applicable for a large cohort of patients. The last project I'd like to talk about is, as Ben mentioned, for optogenetics. So I'm a founder of a startup company, Vedere, with another Berkeley professor, Ehud Isakov. And we sold Vedere to Novartis a few months ago. And hopefully Novartis, which has a lot more resources than our small startup company, will take the optogenetic therapy all the way to the clinic in the next 2 years. So I'll explain how the basics of the optogenetics works. The optogenetic idea came not from me. It came initially from seeing the people at the companies that were making electronic retina chips. And the way the electronic retina chips like Second Sight and the several German companies work is that an array of electrodes are placed on the patient's retina, and that these electrodes stimulate the ganglion cells in patients where they no longer have photoreceptors and they're blind because they no longer have photoreceptors. And what was remarkable about those studies is that it showed that after your photoreceptors have degenerated and you have very low light perception or you're completely blind, the inner retinal neurons still function and they're still connected to the brain. And that's how those electronic prosthetics work, is that they use a video camera and they use an electrical signal, and the electrical signal stimulates the ganglion cells in your retina. And so the way the normal retina works is that you have photoreceptors that are light sensitive. The photoreceptors send a chemical to a relay cell called a bipolar cell. That cell does another relay to the ganglion cells, and the ganglion cells send their signal through your optic nerve to your brain. And so the electronic prosthetics showed that most of the circuit is still there to relay the signal, even after you've lost vision because you no longer have any photoreceptors. And so the way optogenetics works is that it's a gene therapy, but instead of putting back a normal copy of a missing gene to the patient, or like in the case of the dominant disease, trying to edit the defective gene, the optogenetic genes therapy uses a virus to add a new gene to retinal ganglion cells to provide a function that they never had normally. Normally, ganglion cells don't respond to light at all. They listen to the light signals from photoreceptors. But the therapy that optogenetics is using is to add a new protein. In our company's case, it's to use cone opsin, the photopigment that are in cone photoreceptors, and to put the cone opsin in retinal ganglion cells. So the idea is to do an intravitreal injection to inject many millions of virus particles to put cone opsin in retinal ganglion cells. And this certainly is never going to work as well as the gene replacement therapy because we're using a cell that's not normally the photoreceptor. But in our studies so far, it can restore vision to animals that have no photoreceptors, that are completely blind, to the point where the animals can move around and that they can do this in normal room lighting. So we think for AMD patients that have a large defect, a scotoma in their field, or in IRD RP patients that have a large area or a completely blind area where there's no photoreceptors, the optogenetic therapy should be useful. So we now, thanks to the RD Fund, have started a second Vedere 2 company to design more therapies, to design better vectors. And we're hoping that Novartis will provide the horsepower and the funds that they have to take the initial optogenetic program all the way to the clinic in the next 2 years. So I think I'll stop there and I'll give it back to Ben.
Great. Thanks, Dr. Flannery. Thanks for that great overview. And thanks for addressing a lot of the questions. We're a little ahead of the game, but with more than 60 questions, we've got a lot more in the queue. So I am going to stop sharing my screen and we will hand things over to Dr. Adamson. Dr. Adamson, you can share your screen. I'm going to give the folks a brief introduction. Dr. Pete Adamson is Vice President of Janssen Ophthalmology Disease Stronghold area. Prior to his current role, he was Senior Vice President of Ophthalmology Research and Development at ProCure Therapeutics, an antisense oligonucleotide company specializing in the treatment of rare inherited ophthalmic disease, and led the development of sepofarsen for LCA. So take it away, Dr. Adamson. And I think you're on mute.
Okay. Okay. Yeah. Thanks, Ben. Yeah. Just like John, I was looking at the questions, and I think the questions come up a number of times. Why do we work on the genes that we work on? And unlike the other panelists, I'm really the face of big pharma, and ultimately it's us that really need to drive it to launch. But I'll definitely share some of the key elements. I mean, one, we really need a sizable population, and that's not really for commercial reasons, but you need a certain population of people with a disease just to be able to do a clinical trial that would be approved by regulators. That's the first thing. The second thing is you need patients that have some viable retina, and there are situations where people are born with particular IRDs where they don't have any viable retina. And the other thing is you want the disease to progress relatively quickly. I know that sounds pretty awful, that nobody really wants to see this disease progress, but from a clinical trial perspective, you need to be able to treat these patients and see in a reasonable length of time whether your therapeutic is actually effective. And that might be in the 9 to 12 to 24 month time frame. And the other element that I think is worth noting is it costs around $300,000 to study one patient in a standard clinical trial for an inherited retinal dystrophy. And so you can see that once you start adding lots and lots of patients, that gets to a pretty big number pretty quickly. So today I wanted to really jump straight into the clinical and share with you today really the results of a collaboration with Mira GTX, a London-based biotech company. And if I have a little bit of time at the end, I want to talk about our recent acquisition of the Hemera asset, which is an AAV2 soluble CD59, which we'll use for a non-rare disease now, and for a really, really big condition called geographic atrophy AMD, which is the late stage of AMD. And probably between Europe and North America, probably accounts for about 2.6 million patients. So on the first slide, if I can get it to move. Yeah. And some of the other speakers have already said about this, and really what is happening in retinitis pigmentosa and how can we deal with it? And so what I'm showing really here on the screen is, this is a retinal sensitivity map in the center there. And really what it is the physician that tests you, they will shine a light at various parts of your peripheral retinal field, and they'll shine the light at various brightnesses, and they'll ask you whether you can see it. And you'll press a button to see you can see it or you can't see it. And really what they then create is this map. And this map is a little bit like a map that you would take out if you were going hiking in the forest or the mountains. And you can see from that map how high a hill is. And from that map, you can actually make that into an actual hill, really. And this is what we actually call the hill of vision, the hill of retinal sensitivity. And we often talk about this V30. So this is the hill of vision, basically with 30 degrees of eccentricity, 30 degrees of distance from the center point of your eye. And this is a very important measure because it really, particularly in retinitis pigmentosa, which is a disease of the rods, which has already been mentioned, but ultimately a disease of the rods leads in your midlife to a disease of the cones. And, we'd like to be able to protect against rod loss, but it's very, very important as well to protect against cone loss. You can live a perfectly normal life if you maintain that central macular function, which is dominated by the cones. And so, so we use this situation called automated perimetry to assess the rod function at these peripheral parts of the retina. And we also use something called microperimetry, where in the center, where it's a little more cone dominant, where we can look in very, very fine detail at how sensitive your retina is. Now, in X-linked retinitis pigmentosa, this is a relatively severe disease. So most of these patients have relatively good central vision, But in RP, what happens is the rods start to die, and the rods start to die really from the outside coming in, and they start bearing down on that central macular area. And really, we want to try to stop that. Now, you can lose function in RP, lose rod function, in fact, cone function, but it doesn't mean the cells are dead. And if one can reintroduce a wild type gene, a normal gene, then there's a reasonable possibility that not only can you stop visual loss, you may be able to improve visual loss. And so we set out using an AAV5. AAV5 is just a subtype of the viruses that have been talked about today. And in XLRP, the defective gene is called RPGR. And in a gene therapy protocol, we develop the virus, we make sure it does everything it needs to do preclinically, and ultimately we need to go and try it in patients. So we take patients, we try generally a very low dose, so huge safety coverage. Then we move to an intermediate dose, and in this case, a higher dose. And what we look for is really just initial tolerability, so-called phase 1 studies. And then we determine if the high dose is very well tolerated, we may go to a higher dose. Now, what happens particularly with adenoviral gene therapy is when you make these injections, subretinally tend to be less impactful than intravitreal for the reasons that Dr. Boye was saying. But you do get a little inflammation, and so we monitor that closely. And for those patients that do end up with a little bit of ocular inflammation, we treat them very aggressively with steroids, and that tends to resolve. So really the outcome of this phase 1 study was that we saw a good tolerability in the low dose. We saw good tolerability in the intermediate dose, but the high dose, 2 out of 3 patients had some intraocular inflammation. And that's not just a problem from the patient from a perspective that we can treat that, but when you get intraocular inflammation, it quite often limits the efficacy, limits the value of that particular approach. And so really what happens now is you dial back, and I'll show you today the data that we've established today on really the intermediate and the low dose. And in terms of finishing out this, we go through this dose escalation phase, and then we took the intermediate dose. This is a dose that we thought was effective but well tolerated. We confirmed that dose in an additional number of patients. And then the idea now is that we can move into this expansion cohort. This is something that we'll negotiate with regulators, ultimately to agree with them a protocol and to agree with them a set of outcomes that would lead to approval and marketing of the medicine. So in terms of the clinical safety, this is a very usual type of outcome in this situation. We do get what's called adverse events. These were very normal in terms of a subretinal gene therapy. There were so-called 2 SAEs, serious adverse events. One was a retinal detachment, which was related to the procedure and resolved without sequelae. That's not really a worry. And there was one incidence of panuveitis, and I'm going to exclude that patient. That is something that can happen. I mentioned in the higher dose, this was more likely, but this one patient did get panuveitis. There were no dose limiting events. In other words, we could continue to dose the patients as we needed. Okay. And really, this is the type of results that we get. And so what we measure here is the mean retinal sensitivity. How well does an area of your retina sense light? And that's measured by this mean retinal sensitivity that we measure in decibels. And you can see here that in the low cohort, the patients that were dosed with the low concentration of virus or the low dose of virus, there was an improvement, 0.76, but the intermediate did better, 1.05. And if we look at that central hill of vision, we saw again that the low dose cohort, that they did reasonably well at 1.1, but the intermediate did better. And what's also notable and supports what I said a little earlier is in the high dose, where we do see intraocular inflammation, that tends to negate the therapeutic effect of the treatment and really didn't work at all. So really, this is the data. This is really the money shot in terms of if you look at these patients and you look at these patients over various time points. What's been done here is the static perimetry, the V30 hill of vision. So this is how big that mountain of vision is for you, and you want that mountain of vision to be higher and wider. That gives you more area under the curve, and you have more retinal sensitivity. And you can see that in the red is the treated eye, and only one eye was treated in this patient population. And you can see there, as low as 3 months later, post-dose, there was an improvement in this hill of vision. If you look at 6 months, there was a further improvement, and at 9 months, an even further improvement, and at 12 months, an even further improvement. And you can see that the blue line, which is the untreated line, that actually goes down. Now, that's not a reaction to the therapeutic. That is just the natural history of the disease. One expects these X-linked RP patients to get worse. One of the reasons, and as I said before, one of the reasons these subjects were chosen, this indication was chosen, is we know what the natural history of that disease is. We know these patients get worse, and we know that these patients get worse over about a year time frame. And so it's easier to plot an improvement versus the untreated eye. So that's one thing. And so if we then look at, so that was using the automated perimetry, that's looking at the hill of vision. But what if we look at the center of the eye, the macula? And for this, we use microperimetry. And this is at the intermediate dose cohort at month 12. And really what you can see there is in the untreated eye, and these are sort of heat maps of sensitivity. But the important thing here is if you focus on the untreated eye, which is to the right-hand side of the slide, you can see that we have a number of values there, and they're measured in decibels. And a negative number is it's not a bad outcome, but it's not an improvement. It's a reduction in sensitivity. And so that's what you'd expect from the untreated eye. That's what we saw. And so across this patient cohort, we've shown that there's been an improvement in the peripheral or the automated perimetry, the more peripheral perimetry. But here in the microperimetry, we can show that if you then focus on the treated eye, you can see that 3 out of the 4 patients had a really significant improvement in the microperimetry. And even the patient 004 that didn't do as well, it didn't show an improvement, actually showed a reduction in loss compared to the fellow untreated eye. And those are shown at the 12-month stage there in the red letters at the far right-hand side of the slide. So again, just to reiterate, we're showing an improvement here in the microperimetry, the central macular function, but also in the more peripheral retina, more in the rod domain, which is very indicative of a clinically relevant effect. Now, what is obviously very clinically relevant is whether there's an improvement in the patient's ability to function. And this is looking at the functional vision assessment. Now, this is a patient that's at baseline. And this video is running slightly 20% faster than is normal. And you can see that the patient is timed through this maze, and the number of errors that occur is noted. And the patient ultimately gets a score. And that circle defines that the patient is having got an error and then a second error. And so this guy took 61.7 seconds to move through that maze. And albeit in the next slide, which is a patient that is post-dose, but at 9 months, you can see that the patient really walks through the maze pretty unaided, no errors, and much, much faster. And some of you guys may have seen similar data to this that was presented from Luxturna. This is, at least in our view, this is very relevant because we can look at these psychophysical tests, these perimetry and microperimetry, but really perhaps that doesn't mean anything for the patient. What this means is that patient is able to move around, makes sense, the ability to see larger objects, and ability to navigate. And one thing I just want to point out is that although the maze was the same for both of those tests that you saw, the maze has changed all the time. This is done many, many times. And also, very importantly, this is done at different light levels because certainly in some of these patients, if the room is very, very bright, then they can see pretty well. If the room is very, very dimly lit, they struggle much more. And so that's why this maze was done at different light levels. And on this slide, what you can see is the difference in the treated eye and the untreated eye. And we look at the treated eye and the untreated eye by simply masking one eye or the other as they perform this test. And you can see that as you go from the left-hand side of this to the right-hand side, the lines represent lower illumination from 16 lux down to 1 lux. And this would be the range of lighting one might experience in an underground car park during the nighttime hours. So pretty dim light. And you can see that, and this is what would be expected here, is that the change really is at the lower light levels. So again, very consistent with what's thought to be a mechanistic approach for determining an improvement in the ability to actually use your vision to do the activities of daily living. So in conclusion, both the low and intermediate dose cohorts achieved significant improvements in visual mobility at low light levels. There was clinically meaningful improvements in retinal sensitivity, both volumetric and point-wise. And I haven't shown you the point-wise data, but one of the reasons we look at point-wise data is the regulatory authorities have a very strict definition of what they think is clinically meaningful. I can tell you that there was significant improvement in a certain level of change that the regulators would find meaningful. 6 out of 7 subjects in this cohort demonstrated improvement in stability in the treated eye versus the non-treated eye. And basically it looks essentially pretty safe. So this is pretty good phase 2 data. And we now need to negotiate with the regulatory authorities in Europe and the United States and around the world to basically agree the next phase of development. And that next phase of development really is literally an agreement that we need to go out, we need to show the things that they want to see in the way that they want to see them. And we have to come back with an efficacy profile, how well it works, but also a safety profile. And at that point, we will file to be able to market that medicine. So just in the last minute or so, I wanted to move on to really what we've done next. So we've been pretty buoyed by the data that we've seen in the IRD space. And I think it's worth noting that gene therapy really came of age via the eye, via retina. And there's a huge number of reasons for that. One is that the eye, at least the subretinal space, is immunoprivileged, so you don't get as bad an inflammatory response than you might in other parts of the body. One is these things are difficult to make, and we're using very small doses. A rough back of the envelope calculation would suggest that I could treat 50,000 retina patients for 1 liver patient. That's the sort of uplift in the sort of dose you would need. And I think, and the other thing is most of the cells that are the target of gene therapy, ganglion cells or the retinal pigment epithelium or the photoreceptors, they don't turn over. You don't lose them and regenerate them. So a cell that gets a gene therapy and gets the gene within it really is there for the rest of your life. And hopefully you will never need another dose of the gene therapy. But building on that success of retina and we've already talked about the approval of Luxturna, really, and Ben talked about this a little earlier, really retina and the eye is making the next step into the common broader footprint diseases. And he talked about REGENXBIO and Vedere in the wet AMD space. And he talked about Hemera. Now this is the asset from Hemera, and it's actually a soluble CD59, which is part of a very complicated pathway called the complement pathway. And those people that are familiar with AMD will know that you get early signs of AMD in your sort of midlife, and you get what's called drusen. As you get older, that drusen may become confluent, and that's really noted as AMD, early AMD. And then you get sort of intermediate AMD where you start getting visual issues. That can go to neovascular AMD where you grow blood vessels. Those are the things that we have drugs for. Or you can go on to what's called geographic atrophy AMD. Now, geographic atrophy AMD is a bit of a problem because you actually lose cells in the eye, the so-called retinal pigment epithelial cells. Now, a number of companies out there in the space have been using intravitreal injections of proteins and had relatively good results in that space. But we want to go a step further here, and we want to treat patients really in a one and done fashion. So we've got this now. We'll be starting clinical studies sometime in the future. But really, I think it's very, very important now that we can take gene therapy in retina to the next stage and start treating the millions of people with these multifactorial diseases that are not genetically defined, and move that forward. And I think it's interesting to say, maybe the last comment I'll mention is that that's not really possible with subretinal injection. There probably aren't enough surgeons in the world to treat wet and dry AMD by a subretinal injection. It's a pretty complicated procedure. And so this is actually an intravitreal injection. And so I'll probably leave it there. I'll probably stop by just acknowledging all the people that have helped with this, particularly Mirror GTX, our partners here, our participants and families, obviously the clinicians and collaborators, and of course Janssen, who are basically taking the next step here, both in the RPGR space, achromatopsia space, and now back into the AMD space. So I'll hand it back to Ben.
Great, excellent talk, Dr. Adamson. Very enlightening. I really liked the hill of vision description. You know, trying to find ways to measure vision, retinal sensitivity are challenging, and it was great to see how well that person navigated the mobility course after treatment. That's always traumatic. But that also leads me to a first question, really, and I'll direct it toward you, Dr. Adamson, I think first, although the other folks can respond as well. But look, Luxturna has sort of been our baseline for expectations for both being in a clinical trial and gene therapy expectations. And it's sort of a big question, but what do you think a patient should expect when they go in a gene therapy clinical trial? And perhaps comment on the range of potential outcomes in terms of vision restoration versus just saving vision.
Yeah, I mean, I think, you know, in terms of preparing for a clinical trial, the first thing is you want to have the choice. And the only way of getting the choice right now in the inherited retinal disease space is to know your genetic mutation. If you're fortunate is not the right word, but if you happen to know what your mutation is, and I've seen many questions on the stream here about RP patients that have an unknown causality, and right now there's nothing we can do about that. But get to know your genetic defect. That's one thing. The second thing is get on the register like My Retina Tracker, because companies will come to those type of registries and look for patients and advertise for that. I think that so basically you want to have the choice. I think the second thing is you'll get great care during the trial. We are talking here about monogenic diseases, so there's every reason to believe that it will benefit you. Now, ultimately, it may not, and I think we can't predict that. But I think the only downside to getting on a trial like a gene therapy trial is if something else comes along later, you might be excluded from participating in that. And I know the regulators are very hot on that, so we don't get any free passes from the regulators about gene therapy trials. But I think you get great care if you're fortunate enough to have a mutation in a particular gene for which there is a treatment. You should consider it carefully.
Right, and maybe as a follow-on question, if I may, not to keep going at you, but re-injection is an issue in gene therapy. So once you have one eye treated or maybe a second eye treated, will that potentially exclude you from future treatments if, let's say, the injections didn't work? when we're talking about a gene therapy?
Yeah, so there's two parts to that. So the first part is important, actually. This idea of the second eye. So most studies start off by just injecting one eye, obviously for safety reasons. And almost in every protocol, it's the worst eye that you get permission to treat. And sometimes that could be quite tenuous in which one that is, but you're basically only going to do one eye. Normally, what we do in these gene therapy studies is we prophylactically cover the patient with some steroid treatment. And so the idea of going back sometime later, sometime quite a long time later, and re-injecting another eye, it's almost like you vaccinated yourself against this new entity that you're going to bring in. And so one of the things that's now being constructed is this idea of having both eyes treated within a relatively short time, and having that just covered by one set of steroid regimen, such that you have the steroids once, because steroids for your eyes are not great. So you want to limit your exposure to those. So people really are starting to say, well, let's do the one eye now, and let's do the next eye, say 3 to 4 weeks later, and we can just do one steroid regimen.
Great. Thanks for pointing that out. Another question I have, and I want to direct this to Dr. Flannery to get back to optogenetics. And it's actually a question I've had for a while myself, just to get your thoughts on it. But a lot of optogenetics treatments will have glasses or goggles that accompany the treatment to enhance the signal or enhance the illumination of the image coming into the retina. And I just want your thoughts on if that will be necessary for all optogenetic treatments, or do you think we can get an optogenetic treatment that will have the right sensitivity and spectral support that maybe you won't need glasses or goggles?
Yeah, that's a great question. You know, there's several different approaches to optogenetics, and one of the main ways the different ones differ is the optogenetic protein itself, and the different proteins have different properties. When you look at them, there's a principle in physics where you have to trade off between sensitivity and speed. And a few years ago, I was fortunate enough to go to a meeting that Avril Daly organized for Retina International, where we had something where I'd never seen before. We had nerdy basic scientists like me, don't see patients. And then we had clinicians that see patients, but we also had a third of the audience that were patients. So what we did is we asked the patients what they wanted out of different therapies, which was incredibly enlightening. And so one of the things that struck me is that one of the main desires of the patients is for motility. And so for optogenetics, I think what's really important is for the patient to be able to move around the environment, their house or outside. And also, like you say, the goggles have their restrictions. So the different therapies, the first one from Retrosense, which is now Allergan, was very, very fast as far as the vision would allow the patient to move around a lot, but it was incredibly insensitive, and it only worked basically in noonday sun outdoors. And if it was any dimmer than that, there was no light perception. So later therapies now have some range of intensity. The ones that we've been working on with the cone opsins, they work over a range for like normal room lighting, which I'm sitting in now, and it'll work maybe 10 times brighter and 10 times less. But that's nothing like normal human vision, which will work from twilight vision with twinkling stars all the way to noonday sun. That's thousands and millions of fold difference. So the goggles help in that they extend the range of all the therapies. So they can allow the electronics to tune the system to move it to brighter and dimmer. So I think for any therapy, you could enhance it with goggles. You never want to bet against electronics, but some of the therapies now will work over a range of vision that's useful with no goggles. And so the other thing we like to do is we like to try to engineer a therapy that's fast enough for motility. And so companies like Pixar and movie companies have done lots of studies of how fast something has to be in order to look seamless. And so those frequencies, I think, are a good target to be able to get up and move around the room. So like one anecdote I heard from this patient meeting was a woman with Stargardt said, sitting on the couch, she can tell when people come in the room, but she can't tell who it is because she can't recognize their faces. So that's not as hard a target as reading or watching television, but that's an interesting target. Another patient said every time they go in a hotel room that's new, they have to memorize the three-dimensional layout of the room. If they could just tell the doorway to the bathroom, that low level of resolution, that would make a huge difference in their life. So I think the targets for how well the therapy works can be defined by talking to the people, patients.
Interesting. Yeah, as I've personally learned about optogenetics and learned from you, Dr. Flannery, and others, it really gives us an appreciation for how complicated vision is. Movement, the spectrum of light, how fast our retinas have to respond, and to recapitulate that is obviously no small feat. So my next question I'm going to pose to the panel, whoever wants to jump in. I know in some of the talks about gene therapies and the delivery mechanisms of using viruses, dual vectors, and so on, there's been some hints to the future of what gene therapy might look like. But what do you see as the sort of the next generation or two of gene therapies for retinal diseases, maybe beyond our classic AAV approach right now?
Can I take that, Ben?
Please.
Okay, so I think, we talked today a lot about just simple gene replacement, but remember we have genes and those genes make RNA and that RNA makes a protein. And I think what we're going to see a lot more of are therapeutics that target that intermediate RNA, and then also therapies that target the root of the problem, which is the gene itself. So that's going to become really useful for addressing dominant disease, where you need to get rid of expression of a bad protein. And you can do that either by getting rid of the RNA or by addressing that bad mutation in the gene itself. So I think different types of therapeutics are emerging that are going to be really effective. As you mentioned, there's going to be therapies for large genes that are based on dual vectors or gene editing, but I think gone will be the days of being hampered from having a gene therapy just because your gene is too large to fit inside a standard AAV vector. I think our path to the clinic is going to become faster for a lot of therapies because the FDA has sort of seen this movie before, as opposed to, right after Luxturna, there only being one thing to look back on. I think another thing that's going to be important is we're going to have a lot more manufacturing facilities available. Gene therapy is just so hot right now that there's so many more places to get your gene therapy made than there used to be. So I think that's going to help us move forward more quickly. And then, of course, with John's work, we're going to have optogenetic-based therapies for folks that don't know their underlying genetic mutation. The same goes for neurotrophic therapy. And then another area that I think about too a lot is that, there's a lot of broader diseases that ultimately impact the eye in late stage. And I think we're going to start focusing heavily on the vision impairment in some of those diseases. So I see a lot of things happening exponentially.
Yeah, I would agree with that. I think we are currently today in gene therapy, we're probably where we were 30 years ago with antibodies and biologics. When the first antibodies came out, they were very difficult to make and they were slow. And we're right now we're there. That's where we are with gene therapy. They're very difficult to make, very expensive to make. And also I think, in terms of the larger genes that we need to address some of these IRDs in particular, and Stargardt's is a good example, we'll end up with non-viral gene therapy vectors. This is already starting, lipid nanoparticles and the like. It's embryonic right now in terms of its development, but it's going to get there.
Right. Any other thoughts, comments on the future of gene therapy? I guess a follow-up question I have is just commenting on the difference between stem cell therapies and gene therapies, and perhaps if there might be a combination approach someday in the future. I don't know if anybody wants to tackle that. That's kind of a big.
Yeah, I think that's been mooted about the idea of transplanting cells with particular genes added to become sort of cell factories, producing a viable factor, whatever that might be. I think even today, I still think a cell therapy approach is an even more complex product than a gene therapy approach. And I think there's promise there, but ultimately they've got to get a product, and that I actually believe that's the challenge. And there are some other challenges in terms of what will these cell therapies do in terms of what will they fix. If you take, say, dry AMD, will they fix, if you put them back in that scotoma, that hole, will they improve the overlying retina? I don't think anybody knows that right today. And so it's hard to get people to invest in that until that sort of information is available.
Right.
Yeah, I can give one example, Ben, for that. The group at USC in Los Angeles and UC Santa Barbara have had really remarkable success in transplanting RPE cells that were made from patients' stem cells and restoring vision. But in some patients where they have a defect in their RPE, like in Best disease or LCA2, if you were to make stem cells into RPE cells from that patient's cells, you'd give them back the Best disease defect or the choroideremia defect or the LCA2 defect. So in those situations, you'll have to do gene therapy to correct your RPE cells from the patient before you transplant them back in under the macula. Otherwise, you give them back the disease they had in the first place.
Right. Good point.
And then Ben, there's Dr. Ray's work where he's, trying to coerce cells in the retina called glial cells to turn into photoreceptors to sort of rejuvenate the retina that way. So I think there's hope there as well.
Right. Yeah, that's pretty cool.
Mm-hmm.
Grow your own approach. And we'll talk about this in a moment, but stem cells is our next webinar for March. So, and Dr. Ray is one of our presenters, so we'll be hearing more about that. I know we've talked about this a little bit, but I want to go back to it because it is one of the most common genes and it is one of the most challenging genes, and that's ABCA4 because it's the Stargardt disease gene. And I'll sort of just throw the question out generally about approaches, or if you want to be specific about what's happening to address ABCA4 from a gene therapy standpoint. If anybody wants to take that on.
Sure. I mean, I know that there are some dual AAV vector approaches being tackled. So very similar to what I showed for myosin 7A USH1B. The dual AAV vector approach is being pursued for ABCA4 by AGTC, as well as, I want to say Biogen, maybe Nightstar, at least a couple of different companies. So the thing with dual AAV vectors is if you go from a single AAV delivering a gene which makes a protein to two AAVs that deliver halves of a gene to make a protein, you're taking a hit in efficiency, right? Because you need to get two AAVs into the same cell. And so that can work well in some indications where the level of therapeutic protein expression that you need in your retina is relatively low. And we're fortunate in USH1B that the levels of full-length myosin 7A needed are not very high. The challenge with ABCA4 Stargardts is that we need a lot of ABCA4 in our photoreceptors to get the job done. So I think right now the emphasis is being placed on optimizing those dual vectors to make sure that they drive full-length protein expression most efficiently. But that's one strategy definitely that's being heavily pursued. I'll let the others chime in if they know more.
Yeah, I think the thing with Stargardt's as well is it's a photoreceptor disease which then becomes an RPE disease. And so I happen to know that Iveric, that have got a complement inhibitor, they're actually doing a clinical study in Stargardt's right now. I'm not quite sure how that's going to read out, or really, in fact, what they're measuring. But it's interesting that I think what they're trying to do there is to control the RPE cell loss, which is something that happens downstream. Whether that translates into a real patient improvement, we'll need to wait and see. But at least research is happening in that space.
Right. And I think an important point to add on is there are a lot of emerging therapies in clinical trials for Stargardt disease. They just don't happen to be gene therapies at this point. They're targeting survival of the RPE cells by trying to reduce the accumulation of the toxins that cause degeneration of the RPE cells. It's interesting because Iveric Bio, as you say, are looking at this protein, but then they have this whole other space of preclinical gene therapies. So they've kind of got two camps going.
Yeah, and Iveric is also pursuing the mini gene approach, right? So seeing if they can take out some unnecessary elements of that gene and squeeze it down to a size that it can fit inside a regular AAV vector.
Right, right. And I know Hemant Khanna, I think at UMass up in, well, obviously Massachusetts, is pursuing that effort as well. We had him at our Visions conference, and he talked, our virtual Visions conference, he talked about that quite a bit as well. I don't know if anybody wants to comment on people who are having a hard time having the gene mutation identified for their disease. This is getting a little off topic from gene therapy per se, but it comes up a lot in just about every educational webinar we do. People may have been recently tested with a large panel and they get inconclusive results, and things like gene therapy may seem a little less accessible. I don't know if anybody has any thoughts on just how people should think about proceeding if they haven't been able to find their or their loved one's mutated gene.
Yeah, this is a huge barrier that's well recognized, and there are some countries in the world that will reimburse genetic testing for inherited retinal disease. The Netherlands is one I happen to know, but I also happen to know the United States is not one of them. And so really, you know, as I mentioned earlier, you really do have to find out what your mutation is. As more gene therapies become available, as more bigger companies start to develop them to filing, then these companies will help you find out what your mutation is. I know that we have plans to do exactly that, to create a registry, to actually create a concierge service such that you can find out your mutation, you can be taken through the process. And I think that's a natural evolution of bringing these types of therapies to market. But I think, you know, in the medium term, see if you can get your ophthalmologist, your physician, to direct you. It could be My Retina Tracker, or it could be something in country, or to an academic center that's interested in having and finding out your mutation for research purposes. I think ultimately, though, I think you have to push for it.
Right, that's an important point. You have to advocate. And I'll add, we didn't cover this today. We often cover it, but the Foundation Fighting Blindness has a no-cost genetic testing program that most doctors, eye care professionals in the US can order a no-cost genetic test, genetic counseling, through Blueprint Genetics. It's a good panel. Now, if you've recently been tested with a large panel, you may have to wait. But if you haven't been tested for a few years, that's a great option. If you go to fightingblindness.org, you can learn more about the program. The bottom line there is you go to your doctor, you get a clinical diagnosis, and it's the doctor who orders the test from Blueprint Genetics. So we have about 5 minutes left, and we are going to turn it over to our closer for this webinar, Renee Paulsell, our National Director of Chapters. And Renee?
Thank you, Ben.
Take it away.
Thank you, Ben. Appreciate the introduction. First of all, I want to thank all of our speakers today. It has been a remarkable presentation, and we certainly appreciate the panelists who've joined in. Questions that weren't answered, we'll work to get those to you. You're going to be getting a survey post this call for those of you that have registered, and we will be sending out a video recording of this, so you will have that so you can watch it again in its entirety. And we'd also like to hear from you and know who you are and where you're listening from, so that we can continue to make sure that you're included in all of our mailing introductions to these and other important programs that we're presenting. On the screen, what you're seeing is a slide that is talking about our Hope from Home. Our Hope from Home, Davida did such a wonderful job in our introduction. And again, thank you to Davida Luehrs. Davida is serving on this wonderful committee along with countless other volunteers nationwide. Sunday, February 28th is Rare Disease Day. And so we at the foundation are hosting Hope from Home. This is a virtual gala with lots of fun. Kevin Nealon from Saturday Night Live fame is our host. And we also will have a silent auction, a live auction, and lots of fun that evening. So again, on the screen is the information about how to get involved in that. And that program will be beginning at 7 p.m. Eastern Time on Sunday, February 28th. Also, we are excited to talk a little bit about some of the things. If we can show the next slide on our screen, our next national chapter webinar, as Ben alluded to, is on stem cells, and that is on March 20th. And that begins at noon Eastern and will go for 2 hours, just like this session. And we have a wonderful, wonderful group of speakers lined up, including our own Ben Shaberman, who does such a wonderful job for us. But we also have Dr. David Gamm, we have Dr. Tom Ray, and Dr. Michael Young. And so really an all-star lineup for that. So again, do not delay. As soon as that registration comes out, we definitely want to make sure that you're counted in the numbers for that important national chapter webinar. And last but not least, would you like to find out more? Would you like to find out about a chapter near you? We would love to have you join us on our journey. We would love to talk to you more, and we'd love to have you get involved. So at fightingblindness.org/chapters, you can get information. We will be glad to chat with you. We'll be glad to answer your questions and keep you in the loop in all the things that we're doing here at the Foundation Fighting Blindness. So I do want to thank again Davida Luehrs, our wonderful, wonderful host in the Washington, D.C. chapter. Also, Ben Shaberman, who is just always such a wonderful MC and presenter for us for all of these wonderful national chapter webinars. Kelly Cruz and Chris behind the scenes, making sure that we stay on track and keep this thing running. Again, just takes a village, and we appreciate all of you joining us and for your support of the Foundation Fighting Blindness.
Thanks, Renee. Thanks again to our expert panel, Doctors Flannery, Adamson, and Boye. Dr. Boye, feel better. Thanks for being a trooper. And we look forward to seeing everyone in March. And keep up the great work out there raising money for research. We appreciate it.
Thanks, everybody.
You guys.